Hi, everybody. Welcome to part 3 of the Energy and Enzymes lecture. Oh, there we go. So previously we had talked about ATP, and we learned that ATP is a coupling molecule that will link exergonic reactions and endergonic reactions through the gain or the loss of a phosphate group. And we are going to learn today that that is not the only energy coupling molecule we have. We also have energy coupling molecules called electron carriers. Electron carriers are interesting because they couple reactions, but they do it by donating or taking electrons. So for example, one, what we refer to as NAD plus, will become reduced, right? Think about what reduced means. If you remember back to chemistry, if you gain electrons, it's reduced. So it becomes reduced, it gains electrons to make NADH. And this reaction here is going to show that. We look where I'm pointing, NAD plus is going to gain 2 electrons, and it's also going to gain 2 hydrogens. We won't talk about them much, but that also happens. So NAD plus gains 2 electrons, and it becomes NADH plus a hydrogen ion. I like to think about the NAD plus as a depleted battery, and when it gains electrons, it becomes a full battery, right? NADH is energized thanks to those high energy electrons it picked up. NAD plus is going to be low energy. FAD is the same thing. It's another electron carrier. FAD is low energy, picks up some hydrogens and high energy electrons and becomes the energy carrying molecule FADH2. Here is how this would work in a biological system. So we have an exergonic reaction. What's an example of an exergonic reaction? It could be food, for example. So maybe we've broken down the food that we eat. And we know that because it's exergonic, it's losing energy. So some of that energy lost can be utilized. Some of that negative delta G is going to be in the form of a high energy electron. This high energy electron can be picked up by NAD plus. NAD plus is low energy, but when it picks up this high energy electron, it becomes an energized carrier, NADH. NADH then can go somewhere else in the cell and it can drop off a high energy electron to make an endergonic reaction happen. Right? What would an example of an endergonic reaction be? Maybe like building of glycogen, right? That's a reaction that happens in our bodies. We take glucose and turn it into glycogen. So that is endergonic, so it requires an input of free energy. Where is that free energy going to come from? It's going to come from the electron. And notice that the net reaction is downhill because all of that energy is continually lost into forms that we can't use. Okay, so this brings us to part 2. So we have these reactions and how they proceed. So if reactions are downhill, why don't they just happen spontaneously? We said these reactions are spontaneous, but they're not really spontaneous. So for example, starch is not going to spontaneously break down into glucose, right? We have starch in food and it just stays that way. So it simply digests on its own. We need an input of energy, right? And the reason is because those covalent bonds are pretty stable, so they don't just break on their own. Breaking down large molecules requires an input of energy, right? Similar to trying to burn a log. Logs are going to react with oxygen, but it won't happen on its own, right? Logs aren't going to spontaneously combust. We need an input of energy, and that's called activation energy. Activation energy is the energy needed to make a reaction happen. Those large biomolecules are pretty stable, so if you want to break the bonds of a large biomolecule, you have to input some energy. So that is activation. The amount of energy needed to destabilize the bonds of a molecule. So the activation energy is like this hill that we've got to go over to get to the product side. You may remember this from chemistry. Our reactants to our products is net downhill, right? But we've got to go over this hill before we can get there. We've got to overcome the activation energy first. So it moves the reaction over an energy hill. There we go. Glucose is a molecule that we break down in our bodies pretty and we need activation energy to break it down. But we're not going to use a match because that's not going to work in a human body. We've got to use something else for activation energy. So how are we going to deal with this activation energy? We can't use a match. We have to use something else. We use what is called a catalyst. Again, you may remember this from chemistry. A catalyst is going to reduce the amount of energy needed to start a reaction. In other words, it's going to reduce the activation energy. All right. Here is our normal activation energy all the way to the top and then back down. That's a lot of energy to gain. This is the activation energy required for an uncatalyzed reaction. But if we have a catalyst, notice how low that activation energy is. It's much easier to get over. The new activation energy is a lot less energy. So what does a cell need to do in order to reduce its activation energy? We need something special called enzyme. These are biological catalysts. So we're going to watch a video. I'll post one to the topics page that is going to be an intro to enzymes. But basically, this is what an enzyme do. An enzyme is made of proteins. So every living thing needs an enzyme. That's why every living thing has to make proteins, so they all have ribosomes. And the enzymes are going to facilitate chemical reactions. They're going to increase the rate of the reactions, but enzymes are not themselves consumed. They're going to reduce that activation. But remember, they're not going to change the free energy, that delta G, however much energy is lost or gained by a reaction overall. That's not going to change. It's just the activation part. So from the reactants here to the products, that's not going to be changed by an enzyme. An enzyme only changes the activation energy. And most biological reactions are going to require an enzyme. And this is because we need things to happen quickly in a cell. We can't wait. We need millions of reactions to happen, often all at the same time. We can't wait for these reactions to happen on their own. We need an enzyme to help them happen quickly enough and in a high enough quantity. Enzymes are also highly specific. So this is sucrose here that the enzyme is working on, and only sucrose is going to fit in the enzyme. This enzyme is for sucrose and nothing else. They are very, very specific. In lab, you're working with peroxidase. Peroxidase is going to work on peroxide, but nothing else is going to be affected by that enzyme. There are many, many different enzymes in our cells. All right? And these enzymes control all of the reactions that happen. So some important vocabulary. The enzyme is the protein that is helping to catalyze the reaction. It is this purple thing. The substrate is the reactant. In yellow, it binds to the enzyme. The enzyme substrate complex in the very middle here, this is that temporary association between the enzyme and the substrate. The product, that's just what we're left with, right? The orange and pink thing that came as a result of the enzyme's action. And then the active site. The active site is the part that looks like it was cut out with a cookie cutter. The active site is where the enzyme actually does its work. That shape is really important. There we go. So some properties of an enzyme. Enzymes are specific to a reaction. So specificity is important for an enzyme. There is a very exact chemical fit between the active site and a substrate. That is held by hydrogen bonds and some weaker ionic bonds. Enzymes are not consumed in a reaction. They are not part of the chemical equation. They are not used up. Enzymes can be used again and again and again and again. Enzymes can catalyze thousands or more reactions per second. So the enzyme remains unaffected by the reaction. What can affect an enzyme is the conditions in the cell. Remember, an enzyme is a protein, and proteins have a specific shape. So enzymes don't want to denature. So any condition that affects a protein structure could affect an enzyme, such as temperature, pH, and salinity. So we need our cells to stay at homeostasis so that our enzymes remain unaffected and usable. Any change to homeostasis could affect our enzymes. All right, and we are going to stop there for today. This is part 3. Be on the lookout for part 4, and let me know if you have any questions.